All right, so now we're looking at the adaptations of the leaf for photosynthesis. So the leaf is a structure that is adapted for photosynthesis both externally and internally to carry out this process efficiently as possible. So let's now look at how it is externally adapted to carry out photosynthesis. So from this diagram, you can see that I have a labeled leaf here to your left, to your right, right? And that leaf has some areas that are labeled. First on them is the lamina. So the lamina is basically this broad section of the leaf referred to as the leaf blade. Leaf blade, this entire broad section of the leaf, we call that the lamina. And because it is broad and open and just spread out like that, just this is what it is important for. It increases surface area for light and for carbon dioxide absorption. So the light hits the surface of the leaf. So the broader the surface, the more light hit. It has tiny pores on the underside, so the broader it is, the more pores you have, which means the more carbon dioxide can diffuse into the leaf. It is as simple as that. Two, it is usually very thin, and because it is very thin, it means that light can penetrate right through it. So if it is this thick, light can penetrate through it, but if it is this thin, light penetrate through it right through. So therefore it means that all the cells in this leaf when it is thin will be exposed to the light, and also the carbon dioxide when it is diffusing in there, it has a very short distance to travel, so it spreads very easily. But if it is very thick, let's say for example this thick, some light might reach up to this point, so the cells down at this section would not be getting any light. So being very thin is very important. Also is as a petiole, also called a leaf stalk. And what that does is to basically attach the leaf to the stem. Not only that, but it holds it in a particular position. Different plants have different arrangement of how the leaf is held, but it is always held in a position that gives it maximum chance to what? Absorb light. Cool. So that's what the petiole does. The main rib or the midrib, sorry, the midrib or the main vein actually supports the lamina because you can see it drives, it is forming exactly from the petiole and spreads right throughout. So it offers some structural support to the lamina, to the leaf itself, but the main role it is that it is the main supply of water and minerals to the leaf. And the water and minerals are very important because the water is a raw material for photosynthesis. Now the veins are those smaller branches that branch off from the main vein, and that what it does is to bring the water to as many areas in the leaf so that all leaf cells can actually get some water. Waxy cuticle now is the coating or the covering of the upper surface of the leaf, and as you can see, waxy cuticle, what it does here is to prevent desiccation, prevent drying out, prevent dehydration of the leaf. So therefore, what it does is to prevent the leaf from drying out. And remember now that the leaf is held out there in the sun because the sun provides the energy. So the leaf is going to be spread into the sun. So in order to prevent the leaf from drying out, the top or the upper surface, the epidermis, is covered in transparent waxy cuticle. Finally, the lower surface of the leaf, the under the lower epidermis, contains the stomatal pores, those tiny holes that allow for gaseous exchange. Finally, let's now look at the so now let us look at the internal sections of the leaf. So the internal section of the leaf, as you can see from this diagram here, is basically have different types of cells arranged in different ways. So the most important group of cells right here would be the palisade mesophyll cells. So the palisade mesophyll cells are basically arranged in a regular shape. They are basically upright and they're at the upper surface of the leaf. Also, these cells are loaded with chloroplast. So if you see these little green structures, they're the chloroplast, and they're green, yes, because they contain chlorophyll. So the palisade cells are closer to the upper surface of the leaf. There's only one group of cell that separates them from the top, and that is the epidermal cells, which are coated with the waxy cuticle. We just spoke about that. So these cells basically are there at the top to just cover the palisade cells, and note that because these palisade cells are loaded with the chloroplast simply means that they will get the most light. So light hit these cells almost directly. So they now will carry out maximum photosynthesis. Maximum photosynthesis occurs in these palisade cells. Below the palisade cells, you have the spongy mesophyll cells. Now, these mesophyll cells are actually scattered, irregularly shaped, most of them. They have way less, far less chloroplast present, and simply because the light, most of the light will be absorbed by the chlorophyll up above them in the palisade, but whatever light gets down there, they make use of it just the same with their chloroplast. In addition to that, they are spaced, which means that now when carbon dioxide diffuses in through the stomatal pore, which is down here, the carbon dioxide can now spread between each cell and eventually reach up to the palisade cells. So the mesophyll cells, it's important that they are not packed close and tight like the palisade, but they have space between them so that the raw materials such as water as well that comes in via the xylem over here, water when it comes in must be able now to move out and spread to other cells, right? Spread to the palisade and mesophyll cells, all the cells surrounding. So this is the source, the water source, and the xylem is the water source. The stomatal pores is the carbon dioxide source, and up at the top here, the light is coming in, meeting all the palisade cells. So these cells, palisade cells, have a lot of chloroplasts. They're regularly shaped and arranged in a regular position or fixed position, while the spongy mesophyll are more irregular shaped and are scattered. So the palisade is more organized, while the spongy is more irregular in their organization and their shape. And this both of them carry benefits because the palisades being more organized in their arrangement will allow for maximum absorption of light. No light will escape at the top. All right. Unlike that, no, the spongy have more space between them. So if it were that the palisade was organized like the mesophyll, then some light would escape. But because the palisade is so structured and organized, no light will escape. So that's the main structures and how they actually help to facilitate the process of photosynthesis. So you can go ahead and look on this table. You can actually copy this table into your notes to make for a good reference of information. But before we close off, I want to look at two graphs that talk about this term here that you might not be very familiar with, but you are going to be familiar with it today. And that term is limiting factor. So what is a limiting factor? Let us look at photosynthesis, the process. So the process requires raw materials and it requires certain conditions. Now, anytime either a raw material or a condition is not optimum or not in good supply, then the process will slow up or eventually stop. So when you see we talk about the rate of photosynthesis, we're really talking about the speed. The speed. So if we say that environmental factors affect the rate of photosynthesis, what we are actually saying is that the environmental factors can affect the speed of photosynthesis. Now, how do you measure the speed? You look at the product that is being formed. So if you have a reaction occurring, the whole how you know that reaction is occurring is what is being produced from the reaction. So if we want to know if photosynthesis is occurring in a plant, we test for starch in the leaf. So if you test the leaf for starch and you see no starch, then it means that the plant is not photosynthesizing. But if you test the leaf for starch and you find starch, then it means that the leaf is photosynthesizing. We already know the conditions and the raw materials that are needed for photosynthesis to occur. So the first one we say light, second one temperature, third one water, fourth one carbon dioxide. All right. So for example, when it is getting dark, going to the evenings, you don't expect the plants to be photosynthesizing the same speed as they were photosynthesizing midday when the sun was out in all its glory, right? If we operate the process uses enzymes, you don't expect the plants to photosynthesize at the same speed when it is winter, when things cold, like in Canada, -10, -15, -20, as opposed to when it was 40 degrees. So the temperature will affect the rate. Okay? If water, when it is dry season and water is scarce, you don't expect that the plant is going to make glucose at the same pace like when water is available. Same thing for carbon dioxide. Carbon dioxide concentration actually um, goes is very low. So if you increase because carbon dioxide concentration is basically in the atmosphere, it is about what 0.04. So if I increase water, I increase light, and I increase the temperature, and these water, light, let's say light, temperature, water, all of these are in good supply. And because carbon dioxide now reaches at 0.04 percentage, and it's not going to get any higher than this, then it means that even though water, light, and temperature, they're in a good supply, carbon dioxide after a while going to become short. So because it becomes short now, we say that carbon dioxide is the limiting factor. Let's look at two graphs. When you go to CXC questions, these are some of the graphs that they can put to you. Now, at first, you see this graph, you say, no, what go on here? So you don't understand. It is very simple. So let's look at the first graph over here. So in this graph, it is saying that the rate of photosynthesis is on the y-axis and the light intensity is on the x-axis. So here is your origin at 0. So remember now, rate of photosynthesis, we say that the rate of photosynthesis is talking about the speed. All right. So we're measuring the speed of photosynthesis. So I'm asking a question. At 0, it means light intensity is 0, which means at midnight we are talking about dark as midnight. Do you expect the rate of photosynthesis in the plants to be anything beyond 0? No, it will be 0 the same because there's no light energy to drive photolysis, which means you won't get any hydrogen atoms to reduce the carbon dioxide to make the glucose. But however, as we continue now to increase the light intensity, so we increase in the light intensity, we expect now that the plants start get light and start to produce glucose. So the rate or the speed starts to increase proportional with the increase in light intensity. Now we continue to increase. We're going to reach a point. When we reach this point, let's say that this speed is 100. So this is the maximum speed that the plant can make glucose at, right? What that is saying is now we increase in light And if you continue to increase the light, no matter how much more light you put, the plant still not making glucose faster than 100. That is saying that carbon dioxide or the temperature is is going to be limiting the process. So what that means, taking the case of carbon dioxide, we know that the concentration of carbon dioxide either needs to be increased as well in order for the speed of photosynthesis or for glucose to be made to be increased. Or maybe the temperature needs to be raised up as well so that the enzymes can work faster so that the rate of photosynthesis can continue to increase. Look at this graph over here. This graph is showing 3 experiments. 1, 2, 3. Now in experiment number 1, it was done under a temperature of 25 degrees C and a concentration of 0.04. Now, when everything was at all 0, and then we start to increase it, we reach to a point where, guess what? No matter how we increase it, this speed of photosynthesis will not go beyond this rate. Let's call this rate A. Alright? Now, note that the experiment was done at a specific temperature, which is 25 degrees C, and a specific amount of carbon dioxide or concentration, which is 0.04 degrees C. Now, the light intensity has always been increasing. So now when you reach to this point, either the temperature need to raise up or you need to increase the amount of carbon dioxide so that the rate would continue to go up. But because these two stop now, both of them now act as limiting factors to the rate, and them make the rate can go no faster. Okay? In experiment 2, note that it was done at a lower temperature with the same amount of carbon dioxide. And because it was done at a lower temperature, so the carbon dioxide concentration in experiment 1 and in experiment 2 are the same, but the temperature is lower, which means that the rate is going to max out at B, which is earlier than A, which means that temperature is the limiting factor here. If we increase the temperature up to 25, then the rate will go increase, go further up to A before temperature and carbon dioxide becomes the limiting factor again. In experiment number 3, the temperature in experiment number 1 and number 3 is the same, but please look good. The carbon dioxide concentration has been increased, and that is why now when we look at this experiment number 3, the speed max out at C, which is significantly higher than A and B, which meant that now carbon dioxide was the limiting factor, because the temperature at experiment 1 is the same at experiment 3, but differently now. Carbon dioxide is higher. The concentration is higher in experiment 3. That's why we have a higher rate. And if we increase carbon dioxide, then we expect that the rate would go higher as well. So that is how limiting factors do affect the rate of photosynthesis. And so the fate of the substances produced in photosynthesis is that you know that the glucose is going to be used for energy in respiration for the cell to carry out other duties, and then the excess glucose is going to be stored as starch for later use. In some plants, glucose is converted to sucrose and be transported all around the plant. Well, not in some, in most plants. And then the glucose is also converted to other substances such as amino acids, proteins, cellulose, vitamins, and so on. All right, so that's where we're going to stop for there. In video number 4, we will look on some experiments in testing, in determining the effect of light and so on and chlorophyll on the process of photosynthesis. All right.